Okay, and now we are back with DNA replication in the cell, and we're going to really pare this down to almost nothing, just so that you get the concept and it'll help you understand other disorders. So, most cells have very simple lives. They grow, they divide, they eventually die. And that's your cell cycle. They stick to other cells, they have adhesion molecules. They have simple lives. Some cells don't divide once organ maturity is reached, and we have things like cardiac muscles, neurons, nephrons, and there's a reason for that. Those are too important, or so the body decides to risk errors in replication. So those cells, once your heart grows to its maturity, the cells can increase in size, but there will not be cell reproduction. Okay. Other cells need to be replaced periodically: skin cells, liver cells, cells that line the intestinal tract. Those cells will undergo the process of mitosis, as opposed to sex cells get meiosis. Um, but they need to replicate their DNA for replacement, and that follows an orderly sequence. In most cells, we have the process of mitosis, and that is when DNA is copied and the cell divides. I am not going to have the whole cell cycle here for you. It's not needed. With normal mitosis, the basic thing that you have to come away with is that the parent cell divides correctly. Each cell has the same amount of DNA and genes in the same sequence, identical and correct to the parent cell. So each copy of the script looks the same as the master copy. Okay? Nothing is different. Everyone follows the same script. That having been said, in any given cell cycle, there can be a mutation. Errors in DNA equals mutations. They are changes. That's when the sequence of the base pairs or the genes, something goes wrong. We have, and I alluded to this in class, somatic mutations. They are acquired during the copying or replacement of cells. That normal mitosis in your skin cell, in your bone cell, in the cells that line your intestine tract. They are generally random and only affect some cells. Now, if you have exposures to mutagens that affect large numbers of cells, you will see changes. But think of this as a coffee stain on one page of one copy of a script. Now, if you dump your whole pot of coffee on your script, you're going to ruin it. But one splash. So most isolated mutations are of no clinical significance, but when we mutate a lot of genes somatically, we get diseases like cancer. We get diseases from those somatic mutations. Then you have germline mutations. This happens during sex cell division, meiosis. It affects the offspring and it affects every single cell in their body. We'll get to mosaicism as a concept later, but just know that most of the mutation-based genetic disorders, like Marfan's syndrome, that are caused by germline mutations, affect the entire script. Okay, so that's like the copy is just different. It's written in a different language. Um, we have different types of gene mutations. So those individual genes that are living on the chromosome. So sometimes we have a silent mutation. And even though a base pair is different or missing, there's really no change in the cell protein that it makes. The amino acids can be made from the information that's given. And we have other types of mutations. We have nonsense, missense, insertions, deletions. Back to our script analogy. Silent mutation has no effect. Gene still forms the appropriate protein. Maybe the font is changed or the page color, but the actor's line appears as it is written. It can be translated and spoken by the actor, or the stage direction can be followed. So the correct protein is coded for, even though there is a change in those base pairs. A nonsense mutation is when an amino acid can't be formed because a stop codon occurs. So this is sort of like a line gets cut off, a piece of dialogue is missing. And just for entertainment value,
I can do this all day. Yeah, I know.
that's what the original looks like. I can do this all day, right? If we have a nonsense mutation, it might look like this. Yeah, it gets cut off. Now you can't make the protein, so the protein stops being made. That is a nonsense mutation.
I can do this all day.
Sorry, not trying to click on that. Back to the script. Missense mutation, so the sequence is altered. So we have the right base pairs, but they're not in the right order, and a different amino acid forms, and now you have a new protein. That's analogous to Steve Rogers saying, I could do this all week instead of I can do this all day. So a protein is created. It's just not the same protein. Okay. It changes the meaning sometimes. If it's an isolated gene mutation, it might not really make difference in the health of the individual. But again, enough of them, um, especially if they're in the same type of tissue or cells, could cause a lot of problems. Insertions and deletions, they add base pairs or subtract them, and that alters the coding. So instead of saying, I think I can do this all day, or I could do this all day, you're saying, I think I can do this all day, or do this all day. That's an insertion or deletion. It again changes the protein. And so, depending on what that looks like, how many cells are affected, there could be clinical significance. And that's why we look at epigenetic factors. So, that is that. We are going to cut this off and move on to patterns of inheritance next. That is a heady one. Get a snack and a drink, and we'll pick up there.